Alrighty, everybody. Hello. Welcome once again to the Digestive System Video Lecture Series. This is the Digestive System Video Lecture number 3. And as you see here, we are continuing with the small intestines. Our last video, we ended with the stomach. Now we're moving on to the small intestines. The small intestines is the key organ of the digestive system, and it has two very key functions. The first is the hydrolysis of all three food types: carbohydrates, proteins, and fats or lipids. So once again, hydrolysis is the chemical or the molecular breakdown of these molecules, catalyzed by various digestive enzymes, by hydrolytic enzymes such as amylase and trypsin and lipase. Those are the enzymes that digest each of these organic molecules. So hydrolysis or chemical digestion, certainly a huge function of the digestive system, and most of it occurs in the small intestine. Now, you might recall we began the hydrolysis of carbohydrates in the mouth with salivary amylase, and we began the hydrolysis of proteins in the stomach with the enzyme pepsin. Not too much hydrolysis of fats up until this point, but at this time, starting right away in the duodenum, the first part of the small intestines, we get a bunch of enzymes entering the picture and massive hydrolysis of these organic molecules. Following the hydrolysis is the absorption. So once these molecules are hydrolyzed or broken down to a small enough unit, they can be absorbed. And 90% of the nutrients, so the vast majority of the nutrients, including water, are absorbed here in the small intestines. So the small intestines is a rather narrow, relatively speaking, a narrow tube. It's a very tubular part of the digestive tract. And it's quite lengthy because the hydrolysis has to occur, complete hydrolysis has to occur of these nutrients, and so that takes some time. And then after it's hydrolyzed, the absorption has to occur, and just about the complete absorption has to occur within the small intestines. So the small intestines, huge functions, hydrolysis and absorption. Those are the two biggest functions of the digestive system, and both of those occur here in the small intestines. So certainly the front role, the leading role in the digestive system. So the small intestine is broken up into three regions. The entire small intestine is about 20 feet long. Some books might say 25 feet long. It makes up the majority of the digestive tract. The first portion, about a foot long, 12 inches or so, is called the duodenum. And a lot of action takes place here in the duodenum, even though it's not very long. The second segment of the small intestine is called the jejunum, and that's colored here in purple, and that's in the middle. And the final final region of the small intestine, in fact the largest region, is called the ileum, and that's shown here in a peachy orange color. And as you see at this point, the ileum ends, terminates, and the large intestine then begins. And we'll get to that in a little bit. So the first part is the duodenum, and here's where enzymes and bile mix with the chyme. And we don't see that a lot in this image. We'll see that in a different image. But the pancreas, if you can envision the pancreas, its fish-shaped structure kind of nuzzles in. The head of the pancreas nuzzles in to this C curve in the duodenum. So the pancreas is right here. Also, the stomach, of course, is right here as well. That's not shown. The stomach empties into the duodenum, so certainly the stomach's here. And the liver. So you got the liver here, the stomach here, and the pancreas here. Even though they're not being shown, they're all around this part. And so a lot of chemicals, the chyme, pancreas juice, liver juice, bile, entering the small intestines at this point. So the enzymes in the bile mix with the chyme at the beginning, the start of the small intestine. It only makes sense. Therefore, they can work their chemistry throughout the length of the jejunum and the ileum. The sphincter of Oddi, also called the hepatopancreatic sphincter, regulates the release of this stuff that we just talked about. It's not shown in this image, so we'll speak of that in just a moment with a better image. Also, Brunner's glands are in the duodenum, and they secrete a basic mucus which neutralizes the chyme. So we have to take care of that pH consideration, that the stomach contents, the chyme is very acidic, and the small intestines do not cannot deal with the acidity from the stomach. So that acidity, the chyme, has to be neutralized. To tell you the truth, I think we already mentioned that the pancreas juice can neutralize it, as well as these secretion from the Brunner's glands. Then we get to the jejunum. It's roughly about 8 feet long, and we get a lot of hydrolysis, so all those enzymes are doing their work, and then we begin absorption. And then the ileum, the longest, lastest part of the small intestine, is where final absorption occurs. There certainly might be some more hydrolysis or the final stages of hydrolysis continuing on, but it's the absorption that is the key and the key point of the ileum. By the time the contents exit the ileum, exit the small intestine, most of the stuff has to be absorbed. So we have to absorb a lot of stuff here in the ileum. Also in the ileum, and we'll see images later, are these structures called Peyer's patches. They're in the wall of the ileum, in the intestinal wall, and they protect the small intestine from bacteria that might be trying to get in from the large intestine. As you may know, bacteria, friendly bacteria, so to speak, inhabit the large intestine, and sometimes they like to creep upward, creep upstream into the small intestine, and that is prohibited. We can't allow that. So the Peyer's patches, these are lymph tissue, lymphatic nodules that kill back bacteria that might be trying to get upstream. So bacteria like E. coli are welcome inhabitants of the latter portion of the digestive tract, such as the large intestine. But these same bacteria, E. coli, to name a key bacterium, is not possible. We can't have E. coli at the front end of the digestive tract. It will cause harm. It can cause serious illness. So we can't have the bacteria that are welcome at the back part of the digestive tract swimming upstream trying to get to the front part of the digestive tract. So here's that image, maybe a little better image to show the interplay between the pancreas, the duodenum, which I guess is what we're speaking of specifically, and the gallbladder liver complex, and here's the stomach as well. So it's a very good digestive system picture. This is kind of the heart, so to speak, of the digestive system where kind of like it's all centered. What happens here is that the chyme exits the stomach into the duodenum through the actions of the pyloric sphincter and the stomach churning. And as the chyme enters the duodenum, it gets sprayed in a car wash-like manner. It gets sprayed with solutions from the pancreas coming in. The pancreas juice, pancreatic enzymes are coming in through this pancreatic duct, and bile is coming down the green duct here from the gallbladder and the liver. If we look at this intense magnification, this is the common bile duct, and this is the pancreatic duct, and we see when they come together here, they form a little tubule here right in the wall of the duodenum called the ampulla. So the ampulla is this common tubule once these two ducts come together. And then the ampulla empties through this bump here, the duodenal papilla. And so bile comes out here, and pancreas enzymes come out here into the lumen of the duodenum, and we can see that back here in this image. So around that opening there, around this opening, is this muscular ring called the sphincter of Oddi, or the more modern, the more recent term, the hepatopancreatic sphincter. So it's a sphincter different than the other sphincters in that it doesn't really regulate the passage of food. If you remember here, the pyloric sphincter went around the whole GI tract and regulated stomach emptying. Well, this sphincter is different. It doesn't go around the whole GI tract. It doesn't regulate really the food at all. It just goes around this little opening, and it regulates the release of these juices from these other organs. So this is the function of this sphincter here, the sphincter of Oddi. The mucosa of the small intestine has a large folded surface. So we see the folds here, these macroscopic folds, which you can see with the naked eye. Those are called plicae circulares. And then on top of the plica are villi, and those are these little finger-like projections. Those are villi. So you see you have a big fold that you can see with the naked eye. Then you have little folds on the big folds. Now villi is hard to see with the naked eye. The interior wall of the intestine might look a little fuzzy or not smooth, but it's hard to really see an individual villus or even the villi themselves. It's more of a texture, kind of a rough, shag-like, fuzzy texture that the villi produce. Then on top of these villi, which we don't have a picture of here, even the villi have little folds in them. So again, you can't really see it here, but the villi membranes are folded. The cells of the villi have folded membranes, and those are called microvilli. And that's also called the brush border because they're so small it looks like a little brush on the cells, and we'll see those later on. Also, there's these things called brush border enzymes, which are other more digestive enzymes that you'll learn about in a short time. Okay, so these folds, these various levels of folding, from the largest folds to the smallest folds, folds on top of folds, levels of folding, basically have the function of increasing the surface area. So the small intestine has a lot of surface. If this were smooth, it would not nearly have as much surface. All of this folding and folding on top of folding increases the surface area, so we get maximum absorption. Remember, to be absorbed, nutrient molecules really have to touch the wall of the intestine. They have to touch these cells. So the more folds there are, the more convolutions and the more surface area, the higher the likelihood of having contact with the nutrient molecules, and hence absorption. You can't absorb the molecule if you don't touch it. So all these folds are designed to touch the molecules more so we can increase absorption. This is the main function of these folds of the mucosa. Notice the folds really are not a part of the muscularis externa. Certain folds, specifically the plica, do involve the submucosa, but the villi and the microvilli do not. There's a photo of plicae circularis, very prominent folding in a jejunum here. More plica. And here we see the villi. So these were the larger folds. Now we're getting smaller and looking at the next level of folding called the villi. And these finger-like structures are the villi. Each individual is a villus. And then additionally, aside from the villi, notice that they have these invaginations. You can kind of see the openings in these dot-like drawings. Those are intestinal glands, or intestinal crypts in the olden days. But notice the villi poke up, and then the intestinal gland pokes in. And both of those play important roles with the small intestine. Now, at first here, at the present time, we're mainly considering the villi here, the villi. So you see here this yellow is lamina propria, and then you see the epithelial layer, kind of this single layer here, and then the muscularis mucosa down below. So we're orienting ourselves here. It's all taking place really in the mucosa. And here's one of those lymphatic nodules that perhaps a Peyer's patch, if we're in the ileum, that's kind of what they look like, and this is their location. Here's a single villus. So just looking at one of those villi, we see the simple columnar epithelium with goblet cells here and there. Also, these brush-like structure, these little hair-looking structures on the apical surface of the epithelium, those are the microvilli. They're not cilia, they're not hairs, they look kind of like them, but those are really convolutions of the cell membrane of these cells. So that goes in and out, in and out, in and out, and it looks like these little hairs because the membrane goes in and out, in and out. Those are not cilia, though. Those go all around the villus, of course. Then within the villi, in the lamina propria, we see capillaries. And the big green thing is called a lacteal. It's part of the lymphatic system, a lymphatic capillary. And the yellow thing is a nerve, which we don't really have to focus on at this point, but it does have a nerve inside of a villus. Let me tell you right now that when the absorption occurs here, so this is where the peak of absorption is occurring within the small intestine on the villi, the amino acids or the glucose molecules, the nutrient molecules get absorbed, they get phagocytized by these cells. So the nutrient molecules have to get to a small enough size so these cells can phagocytize them. And then they're taken in and they're absorbed. Now the breakdown products of proteins, such as amino acids, or carbohydrates, such as monosaccharides, they get absorbed into the blood, into the capillary here, the blue and the red and the blue. The capillary, that's where the proteins and the carbohydrates go. The fat breakdown products, basically the glycerides, the monoglycerides, and the fatty acids, they don't go into the blood directly just yet. They go into the lymph. They go into the green thing. So fats are absorbed into the lymph. Now ultimately, you know, the lymph ends up in the blood, up at the subclavian veins, as you know. So the fats will get to the lymph, the fatty acids, etc., will get to the lymph, get to the blood, but they don't get absorbed to the blood here. They enter the lymph because fats are different. Here's a photomicrograph of the tip of a villus, and it's quite revealing. We see the simple columnar cells with rather abundant goblet cells. And if you look at the edge of the simple columnar cells, you can see that rather thick edge, almost looks like the edge of a toothbrush type of shape. Anyway, that's the brush border. Those are the microvilli. So these cells have a convoluted membrane, and it's appearing as a thick membrane here as the brush border. Underneath the epithelial cells, you see the lamina propria. And then, it's also part of the villus. And then also in the villus is that lacteal, also often showing up as a white, lengthy, long finger-like structure, a finger-like structure, and a finger-like structure. So that's the lacteal. The muscularis of the small intestine is involved in circular muscle contractions called segmentation. This is just the circular muscles contracting, and it squeezes the diameter of the small intestine to enhance absorption. It doesn't move the stuff really in any way. It's just like a squeezing of the walls. So only the circular muscles. The small intestine is not getting shorter. It's just squeezing. It's getting narrower and longer. And this squeezes the contents and enhances absorption. Doesn't move it in any particular direction. There's also within the muscular part of the small intestine is another sphincter. It's called a valve, but it's a sphincter. It's a sphincter by any definition of sphincter, called the ileocecal valve, and this is the sphincter at the end of the small intestine, leading into the large intestine. The cecum is the first part of the large intestine. And here's an image of that. So what we see here is the small intestine, and you can kind of see a good view of the mesentery as well. And then here's the ileum, the last part of the ileum, the last part of the small intestine, and then this is the large intestine, and we see here's a blow-up, and the sphincter would be right around here, the ileocecal valve, regulating the flow from the small to the large intestine. And here's a photomicrograph showing plica in the small intestine. So hydrolysis, kind of review, but also moving ahead. So taking just a half a step back, speaking of hydrolysis, this is when the enzymes break down the nutrient molecules using water. So, for example, starch, which is a bunch of long chains of glucose, are hydrolyzed into smaller chains of glucose by amylase, salivary amylase, for example, in the mouth, in saliva, breaking down the glucoses. When you get to a point at 2 glucoses, still can't be absorbed, but 2 glucoses is called maltose, and you should know that, maltose. So salivary amylase breaks up starches. And pepsin, as you know, broke down proteins, and the proteins were broken down into polypeptides. Not all the way broken down to amino acids, but the big proteins were broken up into smaller chunks called polypeptides. I don't have an image of that here. Now we get to the main players, though. The salivary amylase and the pepsin were kind of just warming up and getting a few molecules started. Now the main hydrolysis occurs with the pancreatic enzymes. And here they are. Amylase, pancreatic amylase that is, trypsin and lipase. And amylase, just like before, digests or hydrolyzes carbs. The trypsin hydrolyzes proteins, or at this point polypeptides, because the pepsin already broke the proteins down to polypeptides. The trypsin now takes over and takes those polypeptides, breaks them further down all the way to amino acids or dipeptides. Very small portions of protein, and then the lipase breaks down lipids into fatty acids and monoglycerides. Ultimately, they don't finish the job, though. The final job is, the final hydrolysis is performed by the brush border enzymes. And these are right on the brush border, right on the membrane of the intestinal cells, and they do the final bond breaking, and then the molecules can get absorbed. So you do the final bond breaking, the final hydrolysis, and at the same time as you do that, because you're right there on the brush border, you can absorb the molecules. So this is why the brush border finishes the job, because the brush border is also where the absorption occurs. So you finish the hydrolysis, and the absorption can begin immediately. And here we see a disaccharide, such as sucrose, is hydrolyzed with sucrase. And we get the components of sucrose, which are an individual glucose and a fructose, and both of those can be absorbed now. Those are small enough to be absorbed, and they are right there, right then, on the brush border. So the brush border enzymes often don't get their fair share of attention, because number 3 takes up a lot of our time and brain space. They're the big players, admittedly, but these finish the job, and if you don't finish the job isn't done. So they're very important in that regard. This picture again. And some cadaver images showing this part of the digestive tract with the duodenum here. You can see the plica, stomach here, pyloric sphincter would be in this region. You see the positioning of the liver, which is probably lifted up some, the gallbladder hiding underneath. So hydrolysis and absorption. That's why we're focusing a little more on these big functions. This is kind of physiology, folks, the big physiology of digestion happening here in the small intestine. So here we get the passage of a bunch of stuff. Nutrients are the big players, proteins, carbohydrates, and fats, as well as water and ions and vitamins and minerals. And it goes from the GI tract, from the digestive tract, from the lumen of the GI tract, into the blood if you're a protein or a carbohydrate, or into the lymph if you are a fat. And we can kind of see that here. If we just look at this first image, this deals with carbs and proteins, and as you see, carbohydrates and proteins, they're both water soluble, so they're treated separately, they're treated differently. The carbohydrates are hydrolyzed to monosaccharides, the proteins are hydrolyzed to amino acids. They get absorbed. We see the epithelial cells here. They should be a simple columnar. They look a little simple cuboidal to me, but pay no mind to that. You can see the microvilli. So here's our best image of the microvilli to date. We see these convolutions of the apical surface of the epithelial cell. And those are the microvilli. Those brush border enzymes are right here on the microvilli. They don't float out into the lumen. They're attached to the membranes. And I've added some stuff in here. Once the amino acids and monosaccharides are absorbed, they go into a capillary within the villus, into the hepatic portal vein. We have a typo there, hepatic portal vein, and then ultimately they're transported to the liver. Fats are over here. They get treated a little separately. The large fat globules, which is what fat forms in water, and this is a watery environment, so the fat is forming globules. The bile salts from the bile change the fat globules into tiny droplets. And we see the purple bile salts have made the big globules into tiny droplets, and then they can be broken down into free fatty acids and monoglycerides by lipase. So this is the hydrolyzation of lipids. Some other stuff you have to know, we'll find out later, but don't mind it now, the micelles and the chylomicrons. We'll learn about those later. But anyway, the triglycerides are broken up into fatty acids and monoglycerides. They're absorbed into the cell, and then you might notice they're reassembled into triglycerides once again. Some proteins are added. We get these structures called chylomicrons, and they assist in transporting the triglycerides through the blood, because the blood is also a watery system, watery environment, so the triglycerides need to be shuttled. They need a carrier molecule, and the chylomicron and these proteins, which form the chylomicron, are the carrier type of molecule that can carry the lipid in a watery medium, which is the blood plasma. And then those chylomicrons are absorbed into the lacteal, which goes into the lymph, ultimately into the blood in the subclavian vein, as I'm sure you remember by now. So we just went over two huge functions of the digestive system, of the small intestine and the digestive system: hydrolysis, focusing here on the enzymes that perform the hydrolysis and their sequence in which they occur. Very important. Know your chemistry. Also, after hydrolysis, the absorption. So getting a little small here, getting a little molecular, and noticing the absorption here at a molecular level. And I think then we have this table to further emphasize all of this. The carbs are hydrolyzed, so this arrow refers to hydrolysis, to monosaccharides, and the monosaccharides are absorbed. These arrows refer to absorption, absorbed into the blood, circulate through the liver. And we go through that with each of these nutrient molecules. So these are the nutrient molecules: carbohydrates, proteins, and triglycerides, or lipids. We talk more about them in metabolism. So this image showing the monosaccharides and the amino acids going into the blood. See, the end of the arrow goes to the blood. And here the fatty acids and the monoglycerides get absorbed into the lymph. So you see the tip of the arrow going to the yellow structure, the lacteal. So the intestinal crypts and intestinal glands are completely separate, kind of. I mean, they still occur in the small intestine, but we're definitely done with those types of topics, hydrolysis and absorption. So now we're moving on to these intestinal glands, or crypts, and they produce the brush border enzymes, which we talked about those already and what they do. But also here's the new part, they produce enteroendocrines. They produce hormones. The cells are called enteroendocrine cells that produce hormone. Entero referring to intestine, small intestine, and endocrine referring to hormones. So this is the intestinal cells that make hormones. And they're in these crypts or glands, which we remember are down here. So the villi poke up. That's where absorption occurs, but these crypts or these glands poke down, and that's what we're talking about now. So the brush border enzymes are made, and actually the whole epithelial cells are made here, and then they migrate upward, surfaceward, and also hormones are produced here which move down into the blood, down below, because hormones are secreted into the blood. So here's the list of hormones and what they do. GIP stands for gastric inhibitory peptide, and here's what stimulates it. For now, we can kind of focus on just chyme, and here's what it does. It tells the stomach to mellow out or decrease activity, and tells the pancreas to secrete insulin. So GIP acts as an antagonist to gastrin. Gastrin stimulates stomach activity. GIP, gastric inhibitory, so inhibit means to stop or slow down, so that's the down arrow here, slowing down or stopping gastric activity. So gastrin turns the stomach on, GIP turns the stomach off. So by being able to turn it up and turn it down, we can regulate it, and that's the whole key here, is to regulate the stomach. So gastrin and GIP regulate the stomach by turning it on and turning it off, turning it up, turning it down. You can't just do one way or it gets stuck there. You can't ever go the opposite way. So you need to regulate something. You need to be able to turn it on and off. So GIP. Also another hormone, cholecystokinin, CCK, also stimulated by chyme, but in this case maybe more of a lipidy component of chyme, causes the gallbladder to eject bile, because the bile is going to help with that lipids, help break down those lipids, help emulsify the lipids, and the pancreas will also start to secrete enzymes. So the CCK is a big turn-on hormone, turning on the gallbladder, turning on the pancreas, and all this stuff then coming in, ejecting in to the duodenum, Secretin, also stimulated by chyme, and is specifically the acidic nature of chyme, causes the pancreas to secrete the bicarbonate ion, which neutralizes the chyme, again helping to raise the pH, bringing it more towards a neutral level, which the small intestine works best under and appreciates. Also stimulating the liver to secrete the bile. Emphasizing here, everybody, that the gallbladder ejects the bile but doesn't make it. So CCK stimulates the gallbladder to eject the bile, but secretin targets the liver and causes it to make more. Different function, similar, both relating to bile, but they are not the same. So know your differences between the gallbladder and the liver. And even though this might be a little scary at first, it can be quite helpful once you get to know it, showing the relationship between these hormones and glands. Enterogastrone is really GIP. So GIP, again, coming from the small intestine, the duodenum specifically, and it negatively affects the stomach or inhibits the stomach. Most of these other hormones are stimulatory, turning various things on. So you see most of these arrows are going up. That's why there's a lot of green arrows here. And the only down arrow here is the stomach activity, and that's the red arrow over here. So try and get to know this picture. It's very helpful indeed. Okay, pardon the glitch. Let's move along. Once again, here's the image of villus and intestinal gland, kind of an artsy image from the internet. In the large intestine, we will discuss in our next digestive system video lecture.